It's about vascular innervation. One can say that the main goal of blood vessels is to perfuse tissues in order to supply nutrients and oxygen. And it's very important that the flow to a particular tissue can be adapted to the actual needs, and this means we need regulation mechanisms. Obviously, the way to regulate flow is to alter the vascular tone, and this means, if you have a look at the vessel wall, we have the circular orientated smooth muscle fibers. If the muscle fibers contract, it will result in a vasoconstriction, and if they relax, we have a vasodilation. Several factors are involved in the control of the vascular tone, and some come from a distance, such as the nerves, and once the nerves have reached the vessels and are surrounding the vessels, we call them perivascular nerves. Some humoral factors like adrenaline, these are vasoactive agents, come from a distance as well. But we have local factors as well. The perfusion pressure directly acts on the vessel wall. And the shear stress does the same thing. I'm not really sure if you all know what a shear stress is. It's a local force which is exerted by the flow on the vessel wall. So if this is the vessel wall, the flow is really touching the vessel wall and is exerting some force. It's called shear stress, and it really influences the vessel wall. And also metabolites produced by the tissues perfused can act locally. But whatever factors are involved, quite often in a combination, it always has to be the endothelial cells and the smooth muscle cells which are involved in the responses. Now, this presentation is completely about vascular innervation. Everyone knows that it exists, but not so many people are aware of the details. So I would like to give an overview of the best known details of vascular innervation. I also have to say there are so many things we don't know yet, especially when it comes to functions. But we know some things, so I would like to show them to you. But it's not so simple, because there's not such a thing as the vascular innervation. The innervation of blood vessels is very heterogeneous. This means that innervation patterns vary among the vascular beds. But this indicates that there is a very high degree of specificity of circulation control by nerves. Well, the varying factors may be types of nerves, transmitter contents, nerve densities, and these may vary with location, which vascular bed in the body, also with vascular diameter, and also there are obvious differences between the species. And during this lecture you will find examples of it all. So I would like to go through the following parts. First, please allow me a couple of minutes and a couple of slides to introduce again the autonomic nervous system, because when we discuss the innervation of blood vessels, it's all about the autonomic nervous system. And I know by experience, also from Holland, that many students find it a difficult subject, difficult to understand, difficult to remember. So I will start with a few slides to set the basics, because I want to make sure that we're all on the same level when we continue to the vascular innervation. Those of you who attended my lecture on the autonomic nervous system on Tuesday will already know that. Those of you who have an excellent basic knowledge of anatomy, especially neuroscience, they will already know it. So if that's the case, please, just enjoy these moments. It's always so great to see something and hear something that you know already. It just takes a few minutes. But I'm sure you all know, so we start with the autonomic nervous system. I'm sure you all know that we can divide the nervous system in two ways. Topographical way, we have a central nervous system with the brain and the spinal cord. We have the peripheral nervous system, which contains the nerves that connect the central nervous system to any part of the body. And we have a functional division. We distinguish the somatic or voluntary nervous system and the visceral or autonomic nervous system. But actually we should combine the two, because we find that somatic and autonomic systems have part in the central and in the peripheral nervous system. You also know that the somatic nervous system is mainly involved and is very important for our contact with the environment. This is a very simple example. We see a glass of water, this sensory input, it reaches our brain, we recognize it, integration takes place, we recognize it from previous occasions, perhaps we realize that we're thirsty and there will be a response, a motor output, we just grab it and we might drink it. In the autonomic nervous system it's different, because the autonomic nervous system is directed inwards, inside the body. It's very important to regulate and coordinate the function of the internal organs in order to maintain the so-called homeostasis. What does it mean? It means it regulates circulation, respiration, digestion, urogenital functions and also the body temperature. But you cannot say that autonomic nerves innervate circulation. You cannot say autonomic nerves innervate body temperature, because they should have a specific target. And there are only 3 potential targets to be supplied by autonomic nerves. First ones are smooth muscle cells, as are found, for example, in the walls of organs, walls of blood vessels, walls of airways. And of course, that's our target for this lecture, because we will talk about blood vessels. But the second potential target is cardiac muscle. It's a specific kind of muscle. And the third potential target is glandular tissue, salivary glands, the lacrimal glands, sweat glands, and so on. But we'll see the innervation of the smooth muscle cells in this lecture. So let's have a closer look at the peripheral part of the autonomic nervous system. So this is all about the somatic part, and this is the autonomic part of the nervous system. Just like in the somatic nervous system, there is a motor part and a sensory part. And when we have a look at the motor part, we see a striking thing, because it has 2 parts. We have a sympathetic and a parasympathetic division. And this means when we talk about sympathetic nerves and parasympathetic nerves, per definition it's motor, it's visceromotor. Sensory sympathetic fibers do not exist. That's the wrong way of saying something. Sympathetic and parasympathetic is always motor. And many organs, but not all, receive both supplies, sympathetic supply and the parasympathetic supply, and it's a kind of a balance in regulating. But we have autonomic sensory nerves, because a lot of feedback is coming from the organs, also from blood vessels, to the central nervous system. But we don't distinguish specific systems within those sensory nerves. We just say we have visceral sensory fibers. These fibers transfer information for the heart, for example. Visceral afferent fibers is a quite common term. But because in most organs you already have a sympathetic and a parasympathetic supply, it is logical to presume that many of those visceral afferent fibers will take the course of the sympathetic and the parasympathetic pathways, but then in an opposite direction. So this scheme summarizes it all pretty well. So this is the central origin, these are the targets, blood vessels in our case. We have a motor supply with sympathetic and parasympathetic systems and a sensory supply. And then it's important to realize that the motor supply is organized in such a way that between the central origin and the targets, smooth muscle cells, there should be a synapse, always. And this means that the fibers on the way to the blood vessels should pass a so-called autonomic ganglion, and there the synapse takes place. In this lecture, I will refer to this ganglia as the peripheral sources. This will be the central source, the ganglia are peripheral sources. Sensory information goes through nerve fibers to the central nervous system. This is the afferent fiber, and the neuron cell bodies are found in sensory ganglia, such as trigeminal ganglion or the dorsal root ganglia. But in the sensory ganglion, there is no synapse. So we have two types of ganglia, the autonomic ganglia with a synapse for sympathetic and parasympathetic fibers, and the sensory ganglia without a synapse. And then the final slide of this introduction on the autonomic nervous system. The sympathetic system originates in the so-called thoracolumbar part of the spinal cord. It includes all thoracic segments and the uppermost lumbar segments. And the parasympathetic division originates partially in the brainstem and partially in the sacral parts of the spinal cord, and therefore we quite often call it has a craniosacral origin. How about the sensory fibers? The visceral afferents may reach the central nervous system at almost any level. It means they may reach the brainstem and the entire spinal cord. Okay, now we come to see the main parts of the lecture, which is about the general morphological aspects. So we'll see where do the nerves come from, what kind of nerves do we have, how about the pathways, something about the terminal plexus in the blood vessels. A lot of work in this respect has been done on the cerebral arteries, and this includes myself. So there will be some focus on cerebral arteries, but general principles can be applied to other vascular systems as well. So we're going to see a lot of morphology right now. First, the sympathetic fibers. Almost all blood vessels have a sympathetic nerve supply. Capillaries don't have a nerve supply, but all other vessels have a sympathetic nerve supplies. Generally, the sympathetic fibers The sympathetic fibers constrict blood vessels because in most blood vessels the alpha adrenoceptors predominate over the beta adrenoceptors. But there are exceptions, and a well-known exception is the facial vein, including its branches, because there's a predominance of beta adrenoceptors. And this means that upon sympathetic stimulation, you'll find the dilation of the facial vein and its branches, which explains the facial reddening or the blushing. The main transmitter is noradrenaline, but there are other transmitters as well. Neuropeptide Y is a well-known one, and they may come in combination, so it is possible that one axon releases two transmitters, which is called co-transmission. It means that one transmitter may influence the action of the other transmitter. Then the question, do blood vessels have a parasympathetic nerve supply? Well, if you consult some major textbooks of physiology, they ignore the existence of a parasympathetic nerve supply. That is strange, because we have known for some time that cerebral arteries and penile arteries have a very dense parasympathetic innervation. The parasympathetics cause vasodilation, and you might imagine for the penis that may be useful under certain circumstances. And for the brain it means you have an additional regulation mechanism. The cerebral circulation should be regulated precisely, and if you have a dilator system, that is very useful. We have several neurotransmitters, acetylcholine, nitric oxides, and also a neuropeptide called the vasoactive intestinal polypeptide. Then how about a sensory nerve supply? Yes, many blood vessels have a sensory nerve supply as well. I'm quite sure you know one example, which is found in the so-called carotid sinus, which is at the bifurcation of the carotid artery, and especially in the proximal part of the internal carotid artery. There you find baroreceptors. These are high pressure receptors. But actually, many arteries, it has been demonstrated for cerebral arteries, coronary arteries, mesenteric arteries, they have a sensory nerve supply as well. And some of these sensory nerves are very special because they are able to release a transmitter at the receptor sites. And I have made a drawing to illustrate that. It's called an axon reflex. And it's a kind of reaction to noxious stimuli, like chemical, mechanical stimuli, extreme temperature. A good example is migraine, because during a migraine attack you will have a very strong constriction of arteries inside the skull. This strong constriction is monitored, it's sensed by the sensory nerve, and the same sensory fibers can release a transmitter at the receptor site, which quite often is calcitonin gene related peptide, sometimes substance P. And these transmitters are powerful dilators, so you can say it's a kind of compensating mechanism. There is a very strong constriction, and this is a way to compensate for this constriction. But at the same time, this information is transferred to the central nervous system and causes pain sensation. So we have seen that many blood vessels have sympathetic, parasympathetic and sensory nerves, and this is especially true for the cerebral circulation, the cerebral blood vessels. And now we'll focus a little bit on the cerebral circulation. So it means I have to introduce the main cerebral arteries to you again. And we know we have 4 supplying arteries, 2 internal carotid arteries, and they are responsible for about 80% of the cerebral blood flow, and 2 vertebral arteries which fuse to become the basilar artery, and this supplies about 20% of the flow. You know that at the base of the brain there's an anastomotic circle of Willis where all the blood vessels come together and form a real communicating system, and from this circle 6 large arteries go to the brain. We have cerebri anterior, arteria cerebri media, arteria cerebri posterior, 3 on the left and 3 on the right. So now we are concerned with those arteries. About the peripheral sources, the sympathetic fibers synapse in the superior cervical ganglion, which is the uppermost ganglion of the sympathetic trunk. Parasympathetic origins there are more of them. An important one is the pterygopalatine ganglion, which is really hidden deep inside the skull. But the aortic ganglion is involved at least in several species, and we know, we've worked on that ourselves, that we have small ganglia in the cavernous sinus, which are also involved in the innervation of the cerebral blood vessels. And when it comes to sensory innervation, it goes through the trigeminal ganglion, and sometimes we talk about a so-called trigeminal vascular system. It only means that the cerebral arteries have a sensory supply through the trigeminal nerve. So let's have a proper look at some of the origins and the pathways. So this is the superior cervical ganglion. It's a very long ganglion, and we did some work on the rats in the past. This is a so-called whole mount preparation. So it's not a section, it's a complete piece of tissue of a rat neck and part of the head of the neck. So, this structure is the superior cervical ganglion, which includes the asterisk. This is the course in the carotid canal inside the skull base, and this is the cranial cavity. When we zoom in, you see many black stained nerves. It's a staining for acetylcholine esterase. You perhaps know that acetylcholine esterase is an enzyme which is involved in the breakdown of the neurotransmitter acetylcholine. So it's especially present in parasympathetic nerves, but it's also present in non-cholinergic nerves, but in a lower concentration, because it has another function. And this staining is a very powerful staining, so we can stain all nerves, sympathetics, parasympathetics, and also the sensory nerves. Because we know that from the superior cervical ganglion, there is a nerve continuing along the internal carotid artery inside the skull. We call it internal carotid nerve. In this case, we follow the internal carotid nerve and we can see how it reaches the intracranial blood vessels and can be distributed over the intracranial blood vessels. Parasympathetic and sensory fibers gain access to the cerebral arteries in the so-called cavernous sinus. The cavernous sinus is very important when it comes to nerve supply to the cerebral arteries, because here the internal carotid artery and many cranial nerves come together. They have a very close topographical relationship. So what is the cavernous sinus? It's one of the intracranial venous sinuses, but it's a special one, because most sinuses are just single venous tubes, just an intradural compartment with an endothelium lining. But the cavernous sinus is different, because when you open it, you find several structures passing through it, like the internal carotid artery, like the abducens nerve. And that can be explained by the fact that it's not a single venous tube, but a plexus of veins. So if you make a frontal cross-section on this level, and we're looking at the frontal cross-section, you will recognize the pituitary gland, the optic chiasm, and you see it is a plexus of veins. The internal carotid artery is running through it. Now, we've worked a lot on this cavernous sinus to recognize and to describe pathways from ganglia to the cerebral arteries. And now we're going to show you a whole mount preparation again in the human situation. And you have to imagine we are ourselves in the position of the sphenoid sinus. And we're going to have a look at it all from this direction. And there it is. So for orientation, this is anterior, this is posterior. This is the very big internal carotid artery which runs inside the cavernous sinus. This enormous nerve bundle is the internal carotid nerve. So it's an enormous sympathetic supply coming from the superior cervical ganglion, entering the skull, and it gives branches going to the orbit, because here is the orbit, for example the musculus dilatator pupillae. But you see it also continues over the internal carotid artery itself to supply the cerebral arteries. This is the ptericopalladine ganglion, a very important parasympathetic source for vascular supply. And you see connecting fibers to a plexus in the cavernous sinus. There's a real neural plexus inside the cavernous sinus containing some small ganglia. And this is the inferior aspect of the trigeminal ganglion. You see several sensory nerves run from the ganglion to the plexus, and through the plexus you can contact the cerebral arteries. So this cavernous sinus is very important when it comes to pathways. Let's have a close look at the ganglia, because with this kind of staining you can even see the individual neuron cell bodies in the small ganglia. In rats we found a similar thing. We were able to do some retrograde tracing, and that proved that these small ganglia are really involved in the nerve supply of the cerebral arteries. So finally, through the cavernous sinus, the nerves reach the blood vessels. And then what happens to the vessels themselves? In the vessel wall, there is a multi-layered organization. This means outside the vessel wall, we have so-called paravascular nerves. They are running in a longitudinal direction and going to more distal parts of the blood vessels. But from there, nerves penetrate the adventitia. This is the adventitia, form a so-called superficial plexus, and from there a deep plexus is formed, which is made up of circumferentially orientated nerve fibers, and they lie against the outermost smooth muscle cells, because this is a tunica media. So here the local innervation takes place. And this local innervation goes through so-called varicosities. This is the cerebral artery of the rat. On these screens it is better visible, but I can only use my pointer on this screen. It's a parasympathetic nerve staining, and varicosities are widenings, bead-like widenings in the axons, and they are the places for neurotransmitter release. Now, if we have a look at those axons, these are axons which are part of the deep plexus, you see that there are many sites of transmitter release in one axon. It's a kind of en passant release of neurotransmitter. And also means, because there are so many sites of transmitter release in one axon, that this contributes to a generalized response. In humans, we found similar things. This is an electron microscopic image of a posterior communicating artery, smooth muscle cells, axons containing synaptic vesicles, and there's a very close neuromuscular relationship, which is very indicative for function, for local innervation. Of course, by this mechanism, you can only supply the outermost layer of muscle cells, as is indicated in this scheme. But you may know that we have so-called gap junctions connecting the muscle cells, and the signal is transferred to other muscle cells through the gap junctions. So that means, in the end, the complete tunica media is the effector when you only supply the outermost layer of the smooth muscle cells. So, have a look at this picture. This is the circle of Willis of the rat, seen from above. We have removed the skull cap, we have dissected off the brain, but the basal cerebral arteries are still present on the skull. And with the light from underneath, it gives a very nice atmospheric impression, as you can see. It would do very well as a painting in your living room, actually, I think. So, the blood vessels are a little bit brownish black because we did the same acetylcholine esterase staining in these arteries. So now let's zoom to this area. And we see internal carotid artery dividing into the middle cerebral artery and the anterior cerebral artery. You'll see there's a very dense innervation. Generally, one can say that the dense innervation means it's a more important control of circulation. For example, we know that in many vascular beds, smallest arteries and arterioles have a dense sympathetic innervation. And that is understandable because in many vascular beds, these tiny arteries are true resistance vessels. It means they are important to regulate the perfusion pressure and also to regulate the local flow. And therefore, in many vascular beds, you will find a dense sympathetic innervation in the smallest arteries. But in the brain it's different because you find the densest innervation in the large cerebral arteries. But that can be explained because in the brain, inside the skull, these large arteries are resistance vessels too. And I will show in a few minutes how this works when you come to the functional aspects. How about the human situation? Yes, we also have a very dense innervation. This is the middle cerebral artery of humans. It's a so-called protein gene product 9.5 staining. It doesn't matter what kind of substance it is, it's just a general nerve stain. So this is the complete sympathetic, parasympathetic and sensory subpopulations around the middle cerebral arteries in humans. So it's very dense, And you might see there is some red and green staining. I always say it's very rewarding to sit on the front row because if you use these red-green spectacles and you have a look, you might see some depth. Is that true? So it means maybe George would like to see it as well. Just try. You might see that this is a superficial plexus, which is more superficial than these deeply situated axons against the adventitial medial borders. Yeah, but she already seen it or not? Okay. Well, I don't have the time to wait until it passed. So, But if you'd like to see, because I have the original picture with me, you can ask me after the lecture and you can have a look. So also in humans, it's very dense. And if I say very dense, is it dense? Yes, cerebral arteries have a dense innervation. Have a look at these comparisons. So this is a marginal artery, which is one of the arteries of the colon, large intestine. This is a coronary artery in human, and this is the middle cerebral artery, which is very densely innervated. So it's true, it's very dense. We did an enormous study, and I will show you the details in a minute, on this innervation of the basal cerebral arteries. We did in the 90s, actually. And a bit surprisingly, the densest innervation was found in the posterior communicating artery. And the posterior communicating artery is this one. So this is the whole mount preparation. On the section, sometimes we found enormous nerve bundles lying against the adventitial, lying on at the adventitial medial border. So it's very strange that this tiny artery is such a dense innervation. On the other hand, it connects two systems. It connects the internal carotid artery system to the vertebral basilar system. So perhaps for some hemodynamic balance, it's an important artery. But we don't know how these nerves functions in relation to this specific artery. When you talk about dense innervation, it's important to quantify, of course. You can have a look and you can have an impression. You can say, well, this is not so dense, this is denser, this is very dense. And in the past, it was quite often judged this way. We call it semi-quantitative. You have a 5-point scale, have a look. Oh, this is average, I give it a 3. This is more, it's 4. But not so precise. So nowadays, we're using computerized image analysis. It means we can, of course, digitize all the images. You do some image processing, and in the end you have a so-called binary image in which all the nerves are white pixels, all the background is black, and then you can simply express nerve density as a so-called area percentage. We did that for red. For red until then, this is also late 90s, it was mainly done in semi-quantitative ways, and they already found that the highest densities in rats were found in parts of the anterior cerebral artery. It means this part. And it was quite often stated in those articles that probably in humans we would find a similar thing. It was automatically extrapolated. So I did an enormous study in the 90s. It was part of my PhD. And I investigated precisely the innervation of all the basal cerebral arteries. So these basal cerebral artery sets were divided in many segments. I stained them as whole mount preparations for PGP 9.5 for general stain, and I'll quantify them. And we found the biggest nerve density not in the anterior cerebral artery, but in P2 and P1. So PCOM is posterior communicating artery, I already mentioned that one. But there is a very dense nerve, very dense supply also in two parts of the posterior cerebral artery, and these are called the P2 and P1 parts. I had 3 groups. This is the normal adult group, an age group, and an Alzheimer age-matched group. And you see there's a tendency for decrease of nerve supply when you grow older. The arrows, by the way, point to statistically significant differences. And a striking fact was that when you compare aged and age-matched Alzheimer groups, there's only one difference in the A1 segments. And that can be explained perhaps as well, because the A1 segment is the first part of the anterior cerebral artery. It runs on the inferior aspect of the brain, and from there perforating arteries go up straight into the brain. And in that area of the brain you will find the so-called nucleus basalis of Minot. It's a cholinergic nucleus, and it's known that when in Alzheimer's disease is one of the first parts which has a serious problem, an enormous decrease in function. So somehow this might be related, because when it's not so functional anymore, it might need less blood. So perhaps this innervation is influenced as well. That was some explanation we had. So this was quite interesting to notice this difference. And it takes us automatically to the third part about functional aspects and also about plasticity. How does the innervation change over the time? So I already mentioned that the sympathetic nerve supply is important to supply resistance vessels. And it does so in the human brain as well. But in the human brain we have a specific mechanism called autoregulation. And autoregulation means it's important to maintain a constant flow when the perfusion pressure changes, because our brain is inside a closed skull construction. It means it cannot handle rapid increases in flow properly. You might have a problem like edema or even intracerebral hemorrhage. So it's very important that in the physiological range of blood pressure, the flow does not change. So you can see it in this graph. This is the blood flow, this is the physiological range, and the flow just is constant. And this means, if this happens, if the total flow is constant, that the tiny arteries can regulate a little bit according to the actual needs of the cortex. So how does it work? So first, this autoregulation is a kind of second line defense, because at the entrance of the brain we have the baroreceptors, and this is a kind of second line effect. We also investigated the baroreceptors. I just show these images because you hardly find any images in the literature, mostly drawings or EM, but not the real morphology. So these are the baroreceptors in the internal carotid artery. But continue with the cerebral autoregulation. How does it work? This is the formula for flow. The flow is the pressure difference divided by the resistance of the blood vessels. And the formula for resistance is as follows. It's 8 times mu, which is the viscosity of blood, times L, which is the length of the vessel, divided by P times R, which is the radius to the 4th power. You can imagine viscosity and length, they do not change quickly. P, of course, never changes. So if the pressure rises and you want to maintain a constant flow, you have to raise the resistance, and this means that you will have a vasoconstriction. And that's very powerful because the radius is in the 4th power in this formula. So that's what happens. If the blood pressure rises, the large arteries constrict as a protective mechanism, just like closing the tap a little bit, because you don't want a flow rise inside the skull. Now the question is, how is this possible? How do these arteries constrict? And probably there are more factors involved, like endothelium, which are producing factors, like a so-called myogenic mechanism. Smooth muscle cells have a very special capacity. They are able to contract as soon as some stress is exerted on these muscles. So blood pressure rise, it means there is some strain on these muscle cells, and they automatically contract a little bit. It's called a myogenic mechanism. It has to do with calcium channels. But sympathetic nerves are important as well, and we think at the moment that the sympathetic nerves are very important at the higher limits of the autoregulation, so they can be very protective in a very severe cases of hypertension. Then they powerfully constrict the vessels in order to prevent the rise of flow inside the skull. Then how about parasympathetic nerves? What function could they have? Well, we got some clue when we found this difference between the rats and humans. So what was the difference again? We found in rats the highest nerve densities in the anterior cerebral artery, in men the highest nerve density in the posterior cerebral artery. And we started thinking, and we realized that the anterior cerebral artery supplies the olfactory region. And you know, the olfactory system in a rat is very important for this species. Not so important for us anymore. But in humans, it's the visual system which is very important. And you know, the visual cortex is supplied by the posterior cerebral artery. So we found some explanation and thought, well, then there should be some common biological mechanism. And we had an hypothesis. We thought, perhaps, if the system is so important, it means that the metabolic activity varies a lot. For example, if I close my eyes, I know that the neuronal activity of neurons in my visual cortex will decrease immediately, and as a consequence, they don't need so much blood at that moment. If I'm looking in this light, there's a lot of information through my retina coming to my visual cortex, requiring a lot of blood. So then the vessels have to open. So we think, if the system is important, if there's an enormous variation in metabolic demands, the flow variation should be corresponding. So it was formulated as follows. The differences in the nerve densities, we call it topographical heterogeneity. It's related to a coupling between the variation in the metabolic demands and the corresponding flow fluctuations in the afferent arteries. Then we're thinking, how can we investigate it? And we found a way to make rats anosmic. You can inject zinc sulfate in the noses, which completely destroys the olfactory epithelium. And this, you can see what happens. This is a normal olfactory epithelium in rat. This is completely destroyed. It was standardized. They had to find a hidden chocolate cookie in the straw. And when they couldn't find it within 5 minutes, we decided, okay, they are anosmic. And then we found that after 4, but also after 2 weeks, something happened. So we found a decrease in nerve density in specifically the anterior cerebral artery, because when you destroy the olfactory epithelium, no sensory information is reaching the central olfactory systems. So the blood supply to this system can be just average and doesn't have to vary so much anymore. And probably the innervation will set itself to a new optimum, which is lower. And that's what we found. When we did the general nerve staining, we found a decrease of, this is the anosmic rat, about 15%. But of course, we wanted to unravel the populations, and then we had a big surprise, because this is a parasympathetic nerve stain, and we found a decrease of between 35 and 40% of parasympathetic nerve supply in only the anterior cerebral artery. So this is the only difference. These are 2 parts of the anterior cerebral artery. The parasympathetic nerve supply is decreased enormously, whilst the sympathetic nerves, they just remained on the same level. So there was no difference in sympathetic supply when the rats became anosmic, but only in the parasympathetic supply. So this was a hypothesis what it means, and we thought perhaps it's as follows. If this is an afferent artery and it divides into 2 arteries, one is going to the olfactory region and the other one is going to another region. If you start smelling a lot, you will need more blood to the olfactory region. It means the flow in this artery should rise, but it wouldn't be so nice if it, as a consequence, you will find a reduction of flow in this artery. You could call this vascular steal. And therefore, we actually should increase the flow in the afferent artery as is done here. And we think that might be the function of the parasympathetic nerve, actually to prevent against ischemia. But if this is the case, it requires a feedback system. There should be some neural reflex. And then the question is, does such a reflex exist? Well, there are indications in literature that we have such a reflex. This is a part of one of the many proposed pathophysiological mechanisms for migraine. But it's the part which is not so doubtful. So we see in migraine there's a strong vasoconstriction, and I explained earlier in this lecture that as a consequence some sensory information is going to the brainstem and you will have a pain perception. Simultaneously there will be a release of CGRP to compensate, there's a vasodilation. But from here there's a reflex going to the so-called superior salivatory nucleus, which is a parasympathetic nucleus, and this gives parasympathetic nerve supply via the pterygopalatine ganglion to the blood vessel, and gives a further dilation. So there indeed is a reflex from the blood vessel over the brainstem back to the blood vessel. That's an interesting thing. But it will be more complex to invent the hypothesis that he just proposed, and we are not that far at that moment. So then, to summarize a few things about functions, we have seen that sympathetic fibers supply resistance vessels. In many vascular beds, these are very small arteries. In brain, these are the larger arteries. Important in autoregulation to protect against hypertension. Parasympathetic and sensory nerves may have some function against, in protecting against ischemia. We know a little bit about more functions, but I leave them out of consideration here. What I did not mention is an important thing about sympathetic fibers. Throughout the body, they have a trophic effect on the vessel wall. It means they're very important to modulate the growth of smooth muscle cells during development, during repair, after injury, or when a vessel is remodeling. And also in the long term, you need the sympathetic supply to maintain the structure and to maintain its function. It's a well-known long-term trophic effect. Also, if you denervate a blood vessel, you will automatically find structural changes. So there is a trophic influence which is important as well. And finally, a function, although I always think it's a bit hard to call it a function, but the nerves are involved in the so-called vascular pain syndromes like migraine and also cluster headache. Then the final part, it's a short part, it gives a little bit of new direction in our research. It's about the relationship between the autonomic nervous system and inflammation. We have seen that nerve densities may alter, for example, during anosmia, during aging, in Alzheimer's disease. And there are a few publications which have demonstrated that on hypertension, you will have an increase of vascular innervation. But you can say those changes are flow and pressure mediated and are just a functional adaptation. You might also find disease-related changes. And it's known that in diabetes mellitus, you will find decrease of vascular innervation. We have found that in peripheral arterial disease, you might find some problems. And now we know that also in inflammation something is going on. And that is related, the last thing is related to the so-called inflammatory reflex. That's something that's only known for about 10 or 15 years, because we know now that the autonomic nervous system is important, very important, in regulating inflammatory responses. As you may know, an inflammation is simply a defense mechanism. It's a defense against something evil. And it's meant to compensate for that. But the inflammatory response should not become too severe, because when it is too strong, it becomes dangerous to our body. So we should regulate this inflammation, and now we know that the autonomic nervous system is part of it. Especially the parasympathetic fibers can really decrease the inflammatory response. And this was the reflex which was proposed and which really is true. Upon the release of cytokines in inflammation, the cytokine release is monitored by afferent fibers in the vagus nerve. The vagus nerve, of course, is an important parasympathetic nerve. It has also somatomotor fibers for the pharynx and the larynx. But many people do not realize that between 70 and 80% of fibers inside the vagus nerve are afferent, visceral afferent fibers. So the vagus nerve is extremely important for feedback from the organs, from blood vessels to the central nervous system. And also inflammatory information, cytokine release, is monitored by the vagus nerve. And then it reaches the brainstem and there's a reflex. Parasympathetic action as a result has the release of acetylcholine, and this can bind to a receptor, and as a consequence you'll have an inhibition of cytokine release. This was proposed in 2009. In the meantime, we know that probably the spleen is somehow involved in this mechanism as well, so it's more complex, but it's a very important thing, a very nice thing, and this may have therapeutical consequences, because if you can stimulate the vagus nerve, and you can do that by implanting a vagus stimulator in the neck, you can perhaps help people with chronic inflammatory diseases. Some minor experiments have been done already in that, on arthritis, successful. So we were able to have some specimens of aneurysms of the abdominal aorta, controls like here, some aneurysm with minor inflammation and aneurysm with heavy, severe inflammation, and these brown structures. And we found out that there are more nerves when the inflammation is more severe. We also did some Western blotting. It's a way of trying to say something about the concentration of a substance. Western blotting of VMAT and VART, these are markers for sympathetic and parasympathetic fibers, and we found the highest concentration in aneurysms with severe inflammation. So probably as a response to the severe inflammation, you will have an increase of the innervation, perhaps as in an attempt to increase the regulation mechanism. So we're going to continue in this because we're interested in atherosclerosis. Atherosclerosis has an inflammatory component, so it's very useful to investigate how somehow by innervation of a blood vessel you can influence development of atherosclerosis. We're working on it now. But there are spin-offs, and this is the final part of the presentation. I'm also collaborating with the oncological surgeons. And when you do a resection of the esophagus, you automatically will destroy both vagus nerves, because when you have a look at this picture, you will see how the vagus nerves just accompany the esophagus, form a plexus around it. This is more detail. So you don't have a choice. You should do a resection of the esophagus because of a carcinoma, so you have to remove the vagus nerves as well. It was found that in those patients that had an esophagectomy, more problems with the lungs occurred than was expected, especially pneumonias, inflammation. So then it was realized that perhaps the destruction of the vagus nerves means that you are destroying the parasympathetic nerve supply of the lungs, and thereby destroying the inhibiting ability of the autonomic nervous system to the inflammation. So then it was thought perhaps we can find a way to spare the vagus nerve branches to the lungs. So one PhD student, he did this project, and first we had to find out the innervation of the lungs properly. So he did many experiments, but also a dissection. This is a posterior view of the trachea. This is the corresponding drawing, and you see these are the large vagus nerve branches to the lungs along the bronchi. And then it was proposed, if you do a resection of the esophagus and you cut the vagus nerve, if you cut it here and here, you will spare most of the lung innervation. But the point is this feasible? So the surgeons tried several operations in our cadavers. It proved to be feasible. And then we checked the remaining tissue for lymph nodes, because it's fine if you spare the vagus nerves, but if you leave lymph nodes with potential metastasis there, then you have a problem. And it was found that not so many lymph nodes were left behind, just a couple of related to the bronchi. So this might give some possibilities for the future. So to conclude, what have we seen? First, the basics. Blood vessels have a sympathetic nerve supply, all except capillaries. Many have a parasympathetic and a sensory nerve supply. We have seen that regional differences are very common. It says something about the function, otherwise you would not have these differences. And we've seen a little bit about the organization. You'll find the perivascular nerves only in the adventitia, where they form a multi-layered plexus. Oh, this is strange. Then we've seen that vascular innervation is functional. It has a role in circulation control. It has important trophic influences. It's plastic. It can adapt itself to altering circumstances. And we know that the autonomic nervous system modulates inflammatory responses. So this was exactly 58 minutes. It was a lot of information in 1 hour, a complex subject of which we don't know many things yet. So I hope that I gave a useful overview of it all. And when you have so much information in a short time, sometimes it may happen that the audience looks like this. But when I have a look at you, it's not so bad. So thank you very much for your attention.